and then you'll learn about human digestion come Thursday, and then you'll have only the lab to do on Friday. So I'm going to go over what the lab looks like, so that way when you get to that day, when you get to the 5th, you will be ready to go when it comes to completing the lab. Okay, and I saw that we have Jude joining us as well. Thank you so much for joining us. And just as a reminder, I'm sure you guys all know this, but just to remind you all that we are going to be doing Mondays, our labs or lessons that go over the lab for the week. So definitely still worth coming in. I highly suggest that you still come on Mondays, because this lab in particular is a little trickier than the other ones that we've had this quarter. So it's a really good idea that you come on Mondays, so that whether it's that same day or whether it's later on, the most important thing is that you are still getting the information you need so that you know what to expect once you get to the lab portion. So like I said, we are going to go over that. So let's go ahead and get started. Now I don't, unlike other classes that we've done together, on lab days, we're not going to do the lab together. I'm going to basically do it. I'm going to be walking through as if I'm doing it myself. And then I want you guys to be following along, taking notes, writing some things down. So that way, when you get to the lab, you can remember, oh, this is when Fierstein did this, or this is when Fierstein said we need to be really careful. So really be paying attention to, the lab and what I go over, okay? So I'm doing two things at once. I'm building the test tube rack. So there's definitely going to be things in your lab kit that you're going to need for this lab, as well as a couple things that you'll need to provide on your own. If for whatever reason you don't have what you need to complete the lab, just let me know and I will do whatever I can to be sure that we get you those materials. So, you're going to need a cracker for this lab, or some sort of cracker. So if you don't have crackers and aren't able to get crackers, let me know. We can figure something out, whether that's me sending it to you through the mail, or ordering Instacart and getting it dropped off to your house, or myself getting it and dropping it off to your house. We will certainly figure it out. Okay, so to start off with the digestion lab, to understand what it is you're going to have to do, I'm going to teach a little crash course of what digestion looks like in humans specifically. So digestion, then you're going to have a lesson about digestion in humans, and then you're going to have your lab. So what we have here are the learning objectives for the lab. So at the end of this lab, you should be able to explain the digestion and waste removal in representative organisms, right? So like the organisms that go through this process. You're going to be able to describe the process of digestion in a human and giving examples from anatomy. So being able to tell me and where all of these things are happening. And the steps, and then identify the individual functions and sites of secretion of digestive enzymes. So being able to determine, which enzyme is used for what when it comes to breaking things down, and when and where this happens. So like, for example, the first step, and we're going to get into it in just a moment, the first step of digestion is actually the minute that food enters our mouth, right? The minute that and maybe initially you're thinking, yeah, duh, when you chew food. But as a matter of fact, the first thing that happens with digestion is you salivate, right? You get the saliva, your mouth starts to water. And you might not have known this, but there's actually an enzyme in your saliva. And that's the first step of digestion. So let's go ahead and take a look at this. Like I said, it seems like a lot. It is a lot on here, but you are going to have a chance to read through this on your own. Okay, so don't get too alarmed if you're a little overwhelmed with all this, because you haven't read the lesson yet, right? So this is like all of the information on one slide. So we're really gonna just spend a little bit of time understanding the basics of digestion, just so we can understand the procedure and why we're doing what we're doing when it comes to the lab itself, okay? So by the time you are completing the lab, remember, you will have learned all of this as well, but at least it's a place to start. So the first step in digestion is you have the mouth. So like I said, in your mouth, the teeth are breaking down the food into smaller pieces. And then you have an enzyme called amylase that in your saliva, that's breaking down the food further. So already your enzymes are at work the minute that you put something in your mouth. Okay, step 2, the esophagus. So now we remember these two definitely get tripped around, and I do it myself. You've got the trachea and you've got the esophagus. Both of those are tubes that go from your throat down into your body. The esophagus is used for swallowing food, right? So what do you think the trachea is for then? What's the trachea going to be for? It's the other pipe, if you will. Sometimes when people swallow and they start to choke, they say, it went down the wrong pipe. Breathing, exactly, Jude, yes. So remember, trachea is for oxygen when you're breathing, and your esophagus is for when you swallow food, right? So over here in number 2, it says muscles in the esophagus push down the food into the stomach. So when you swallow, it's not just that swallowing action, you actually have muscles that are contracting down your esophagus to push it down. The third step in the digestive system is that the stomach gives off hydrochloric acid. Okay, so that's a really high acid, high concentration. And enzymes like pepsin, that break down the food, particularly proteins, and the acidic environment kills many germs. So once you swallow your food and the esophagus pushes it down into your stomach, that's when there's a ton of acid in your stomach. It's really cool because remember, we have specific cells that are created for specific functions, and we have a really thick mucus lining in our stomach that protects that lining from the acid, the acidic nature. So hydrochloric acid is something that's in our stomach, and we produce it in our bodies. And then we have this other enzyme called pepsin that helps break down food as well. Okay, and then once it goes from the stomach, it then goes into the small intestine. So the small intestine is right here. It's this one, it's like that big piece that you think of. So the small intestine, the food from the stomach goes into the small intestine. Good, we missed you. We missed you, Spencer. And then the food moves into the small intestine, which absorbs the nutrients and transfer them to the bloodstream. So this is where when we eat nutritious food, we are absorbing all of those nutrients in the small intestine. It's breaking it down into those submolecular levels. And we're like, oh, this is really good for us. I want this. I want that. And then we put it into our blood cells and it sends it to the body, the parts that we need. We also have the gallbladder. So these are like parts of the digestive system that is a little lesser known, right? So we've got the gallbladder. So one thing that we want to talk about is that the liver, I meant to say the liver. We are at the liver. Okay, that makes more sense. The liver gives off something called bile salts. Okay, so if you get really sick and you've emptied out your stomach, then you know that clear mucousy stuff that you may bring up, that is the bile. So that's like the bile salts that get put into your small intestine to help break up the fats. Okay, so that's what the liver helps do. It breaks up the fats in your small intestine. So that's right here. Now you get the pancreas, which is this guy, okay? This pancreas is, oh my goodness, I can't read my own model. The gallbladder, now we're talking about the gallbladder. So right over here, the gallbladder stores the bile. So now we're making sure that we have the bile from the liver and it secretes it into the small intestine to help break down fat. So it's basically doing it twice, right? The liver puts in the bile salts to break it down, and then the gallbladder takes it again and secretes it into the small intestine to break it down, the fats further. Okay, and then we have the pancreas. So over here, this is the pancreas, right? So the pancreas gives off more enzymes into the small intestine to break down food and adds a neutralizer to neutralize your stomach acid. So it puts in this type of base that's going to help the acid kind of lose its strength in the small intestine. The next step is that any leftover products that are too large or that don't get absorbed through the small intestine and put into the bloodstream moves into your large intestine. And this is what we call waste. So this is where we have solid waste as well. And we have bacteria in our large intestine that's helping us absorb vitamins. Okay. And then after they absorb it, the last thing that you do is you poop. So that's the last step. So anything that your body does not absorb gets put into the large intestine. The large intestine kind of takes out any extra water, and then it moves it into your area that you poop. And then you get rid of the waste. So that was a lot. That's a lot of information about the digestive system in, just a couple of minutes. And so, like I said, you're going to have way more time to go over this when you learn about it in your lesson. And that lesson, I believe, is coming up Thursday. So really quickly, I want to go ahead and move into the actual procedure part. So first of all, are there any questions about how our digestive system works before we move on? Any questions at all? Okay. All right, then that's fine. Then we will move on to the next slide. All right, so what you're going to need for this lab, all right, you are going to need, I have all of it over here. You are going to need disposable gloves. So this is all the things that are coming back or that you have in your kit. Okay, so you need disposable gloves. Okay, so they're going to come in these little bags. There's multiple of them. You're going to want one pair. You are going to need iodine. So remember that it comes in, if you haven't, we did use this once before in Q1. So here is the iodine. If you don't have it, if you didn't open it from last time, you are going to need measuring spoons or cups or bowls. I'm personally using the test tubes because I think it's cool and they have it in the kit. So why not? So they have a test tube rack where you just put it together and test tubes. So you're going to need, and then you're also going to need measuring spoons. And that's really only because we want to be sure that there is the same amount in every container. If you choose to use a bowl or a cup, that's totally fine. You're really going to want to make sure that you have the measuring spoons for that because it's harder to tell, if it's even. And remember, we really want it to be as even as possible because that's an issue with the control, right? We want to make sure that there aren't any variables. So like I said, I'm going to be using the test tubes. You don't have to. I just want to because why not? Okay. Okay. And you're going to need a watch or some way to tell time. And you're going to want crackers. So those are or if you don't have crackers, anything that's starchy like crackers, you're going to want. Okay. So once you have all of your materials, then you're going to need to know your safety precautions. So for example, you're really going to need to be careful, right? We know that iodine is going to, you don't know where iodine is. It should be in the box. I did find it. It was just in this individual bag. And it was kind of hidden in there. So you kind of have to search for it. Now, unless you use the iodine in Q1, in that case, it's going to be wherever you had it last, which I don't know what, that's obviously going to be different for each person. So I'm not sure. where it's going to be for you. But we can talk about that afterwards, Logan. We can always figure something out. Okay. All right. And then, so we're going to have to be careful because we know that iodine stains clothing. It can stain your skin. Obviously, it will come off eventually, but it can leave a mark on there for a little bit in any work surfaces. So making sure that you're putting something down on the table, or if you have like an area where you know that you can risk it, you're going to do that. You're going to avoid eating and drinking. You are not going to want to eat or drink anything during this lab. And you're definitely not going to want to eat anything that has iodine in it. Tell everyone in your household so that they're aware that you're doing the lab, so they don't come in and see, oh, a cracker, and pop it into their mouth. Little did they know it was full of iodine. So making sure, and your spit, because that's part of it. So making sure that everyone is aware of it. And then the iodine. So making sure that you are not eating anything with it or inhaling it. It can also cause burns, so you really just need to be careful. That's why wearing the glove is going to be really, really important. Okay, questions about safety precautions? Okay. Be very careful when you're open, if you do use the test tube. I just got, my test tube broke, so it was just, I got a little pinch. Okay. So this lab, this is probably, of all of the parts of this presentation, this is probably the most important to pay attention to. And that's because this is going to be a little different than previous labs, where previous labs, we were given like a very direct, specific set of instructions. This time, you're going to be choosing what question you want to answer, and you're going to be coming up with your own procedure. So we are going to be writing a lab report with this lab. So you're going to choose between two questions. You're going to choose how pH affects starch digestion, or you're going to test how long it takes for starch to be broken down by amylase. Now remember, amylase is that enzyme in our saliva that breaks food down as we start to chew it. So if you choose how pH affects starch digestion, then you're going to use a different type of substance, either vinegar or lemon juice, and you're going to mix it with your saliva to see how amylase activity is affected by the acidic conditions. Okay, so you're going to use different types of acidic liquids to see how the environment affects the rate of reaction. The other option, where it was talking about how long it takes for starch to be broken down by amylase, you're going to allow the saltine cracker to sit in saliva for different periods of time. So again, we're going to be using our spit. I know it sounds kind of gross, but I actually watched a group of kids when I was in my student teaching in college. I watched this lab of kids doing this, where they actually took liver and their spit and watched it was pretty cool. Watched it actually get broken down. It was really cool until, you have 30 kids who don't really know how to brush their teeth on a regular basis. And then it's not so great because then you smell everybody's breath, and that was not cool. Not necessarily, Logan. So is the knowledge for both of these questions already known? I mean, you could Google it, but it's way more fun if you test it out for yourself. And you can do it differently, so that because it's an opportunity for you to have experience with the material and so it's not just important to know already the knowns, but it's also important to acquire the skills that you're going to need in order for you to better follow and practice the scientific method. So it's not just so much about how to find the answer, but it's how did you get there? What steps did you take? What observation? How did you write your response? Because remember, part of this is also going to be writing a lab report. So not just understanding it yourself, but also being able to articulate yourself and explain and justify your answer is just as important as finding the answer itself. So sure, you could probably Google this, but at the same time, why not do it yourself and find out the answers yourself so that you can get the skills that you need? And then remember that these skills that you learn in science are absolutely transferable to any other aspect to your life. Okay, so good question. It is important for us, it is important for you to test instead of just being taught, because it's part of the learning process. Good question. Okay, so remember that you are choosing your own question. It's one or the other. You don't have to do both. In fact, it's not expected for you to do both, but you can choose which one you want to do, whether you want to know about the acidic environment or if you want to know about how long it takes for things to break down with amylase. So, hi, Christian. This is a lab that's due Friday. So this is this week's lab. We're just talking about the digestion. We're going to be figuring out some stuff from the digestive system. Okay, so there is a general procedure. So when I say general, I mean you should, there's a little note here. It says, do not copy and paste these procedures in your lab report. They must be specific to your experiment in your own words. So the very first thing I want to point out is that this is a general procedure, meaning this is so like it's not specific so that you can apply this to either question. But it is not specific enough where your procedure could be replicated for your experiment. So really important, I'm gonna go over it, but it's really important that you do not copy and paste this, that in fact you add into your own words and write down what you did for your specific lab. So we have the very first thing. So we've got our small saltine. So we've got our saltine cracker, okay? So the first step that you're gonna do, and this is true for both, is you're going to open your saltine crackers and you are going to break it up to simulate chewing, but you're not gonna actually chew it, okay? You are not gonna actually chew it. My dog's about to go crazy because she's sitting in my lap. Okay, so you've got your saltine cracker. You are going to break it up into small pieces and then to get it ready for the test subject. Now, I stabbed myself with one of the, no, Millie. Okay, so here's my test tube. Here's my test tube rack. I don't know if you can see it or not, but there we go. Okay. Can you see my test tube? There it is. Okay. And then I'm going to get another one. I'm probably going to want to create about 3 options because that's typically what is done in science is we have our three options. Okay. So we've got our 3 test tubes, and what you will do is you are going to take it up. So you're going to remove the stoppers. Don't need the stoppers for this. Okay. So you have your test tubes, and then you're just going to break up your saltine as if they were to be in bites, right? So you're going to want to break it up just small enough as if you're chewing it, right? You're not going to actually chew it because that would be gross. Although I guess we're already spitting into the test tube, but still, don't do it. And then from there, okay, you're going to break it up. You're going to break up the saltine into small pieces to simulate chewing. Do not actually chew it. And then once you simulate it, and you're going to want to make sure that there's about the same amount of saltine cracker in each test tube, right? Remember, the only thing that we want to be different is the variable that we're changing. So depending on the question that you are answering, it could either be the time or the environment, right? The acidic environment. So everything else should be the same amount of liquid, the same amount of saltine cracker in each test tube. That's going to be really important. Okay. The next thing is it says to spit into the container. So great. One thing that I can tell you to do, and you need to make sure that you're doing this accurately though, is you need to be, you want to put in the same amount of saliva in each one. Okay, if you run out of saliva, which sounds really silly, but it does happen. if you run out of saliva, what you can do is chew on a rubber band. Chewing on a rubber band activates your salivatory glands and it creates saliva in your mouth. So let me go ahead and do that. This is kind of gross. Okay, so I've got spit in 1 or saliva. Okay, put it in 2. Okay, so I've got my saliva. Probably not you want to make sure that it's going to be the same amount in each tube. So I did not do the rubber band trick, but I should have to make sure that I have the same amount. Okay, the next thing it says is to dilute saliva. So you're going to keep the volume consistent between samples with and without amylase. That's true. I want one without amylase altogether. So let me find the one that is not broken and did not stab me. Okay, so I want my control. So this is the one with no saliva. These 3 have saliva. Okay, so then it says that you're going to, right, you're going to want to keep it consistent. So this is where it kind of changes into your own experiment. So if you're doing time, then the only thing that you want to do is change the amount of time for each one of these test tubes, right? Maybe the control is stays the same for, it should stay the one with no spit should be relatively the same as the other ones, like the longest one. But then what you can do to dilute it is you can add water to it. So you're going to want to add water to it, and then you are going to want to let each one sit for a different amount of time. And you can choose how long each one. Maybe this one is the one that is for, we do 30 seconds, a minute, 2 minutes, right? And then we just let it sit and kind of do its thing. And then after 30 seconds, you add the iodine. After a minute, you add the iodine. After 2 minutes, you add the iodine to see what the results are. Or if you are doing the environment, this is where you have the same amount of saliva, and then this is where you go ahead and you do, like, maybe you do lemon juice in this one. Maybe you do apple juice, and then maybe you do orange juice, right? And then the most important, then water in this one. And the most important is they all have to be the same amount. That's the most important thing about these experiments, is that you are keeping the consistency, right? And then what you'll want to do once you put in the environments, then you're going to want to have the same amount of time for each one. So maybe you do 3 minutes for all 4 of them, and then you pour the iodine to test the different results. The last step is that you don't eat anything. I think that goes without saying. So that's kind of how I seriously have to repeat myself all the time when it comes to the chewing thing, because it's true. We don't eat it. And if there's anything I learned about teaching in the classroom, there's always somebody who's going to eat it. So yes, I repeat myself a lot because someone is not listening. Yep, 101. I like it. Okay, so that's going to be the general procedure. And like I said, depending on the question that you are answering for your lab is going to determine which or how you draw your procedure. But that's the general procedure of this lab, okay? So you're going to want to do something based off of it. Okay. Questions about the procedure. I want to spend some time going over the lab since this is our first official written lab for this quarter. And I want to make sure everybody knows exactly what should go into it and how it's going to be graded. So are there any questions about the procedure before I go on? Okay. So as you're going, okay, so before you start your lab, here are a couple things that you're going to want to consider. You're really going to want to think to yourself, what is my independent variable? What's the IV, right? The independent variable is going to be what you are changing. It's what you are testing, right? So depending on which procedure or which question you want to go with to answer for your lab is going to determine what your independent variable is. And I'm not going to tell you what that is. I'm going to let you kind of sit with that. We've been working with independent variables all throughout middle school. I know I taught 6th, 7th, and 8th grade for 4 years. So I know you guys have learned about independent variables. If you have a question about that, feel free to ask or come to help sessions and I can help you with that. Then the other thing you're going to want to consider is what is the dependent variable? So what are you measuring? How are you going to tell whether your independent variable is making a difference, right? And then what are your control values? So what stays the same, right? So making sure that you, like I said, are keeping the same amount of liquid. If you're only testing the environment, then you want the same amount of time. The same amount of cracker in there, the same amount of saliva in there. So really making sure that you guys are keeping the controls the same. Okay, this is like the big thing, and it doesn't have to be as overwhelming as it may seem. But what you're gonna wanna do for your lab report is there are 5 sections of your lab report. The first section is the title. Now, recall, you are writing the lab report. When you download the lab report off of the lesson, they are going to have like a little quick explanation of what each section is, and then they have their own example. You're going to want to delete that example because you're going to be writing your own lab report based on the procedure and the question that you're answering. So the title, you're going to tell us what the lab is going to be about. Here's a rule of thumb. If you don't know what a good title is, you can always do the IV versus the DV. So the independent variable versus dependent variable. That's a rule of thumb. It's not going to get you all the points, but it's going to get you points, right? So that's going to be the first part of your lab report. The second part of your lab report is the introduction. So this is where it talks about principles, meaning like content, what are you learning about? This is also the point where you state your purpose. So tell me, why are you doing this? Like, what's the big deal of this lab? What can it be used for or applied for in other areas of science? And the lab, if you click through the slides in the lesson, it gives you specific questions that they want you to address for the inclusion. Okay, the introduction, the introduction paragraph. So being sure that you're including everything and answering the questions. But you want to make sure that you're answering the questions in a way that is not obviously answering questions. So I'll go over that in a second. The third section of the lab is the experimental methods. So this is where you describe how the experiment was conducted. You're going to tell me what the controls are and how the measurements were taken. So this is kind of where you would, if someone were to take your lab report, they should be able to read this section and recreate it. So my 2 favorite words are specific and detailed. So you want to be really specific in this one so that you can be sure that you are, that somebody wanted to redo this, they would be able to, okay? Fourth section is the data and observation. So this is where you start to include your data. You really want to be thinking of including graphs. So whether that's a bar graph, whether it's a line graph, you really want to be including some sort of visual representation of your data. Part of your experimental methods, I should also mention, so let's go back to the previous section. For the experimental methods, you are going to want to include a data table that you use. Okay, and then in the data and observations, you want to include a graph or some sort of data table if that's what you used. You want to include that and you want to describe your results. So you want to not just like, when I put my saliva in the test tube, I saw the saltine cracker bubble. That's great. That's one data of observations, but go a little bit farther. What do you think that meant then? If you saw something bubbling, what does that typically mean of a reaction? And what you know about what we're testing, how does that relate? So really for number 4 is like, 3 is like, this is what I did, this is what I got. 4 is like, this is, the data that I got and some things that we can connect with it. Then you've got the last part of your data, and some would argue probably one of the more important ones, because this is your discussion and conclusion. So now this is your opportunity to take your data and take your introduction and put it together and connect it. So what did your data tell you about this lab? Is your hypothesis correct, right? So telling me about specifically how your lab connected and how it answered the overall question. Now, the lab lesson is going to give you the questions that they want you to answer in your conclusion. Now, the only thing with this, however, is I really want you guys to get into the habit of embedding the answer. So not just saying, they'll have questions for you. And I don't want you to just rewrite the question and answer the questions in like one answer at a time. The paragraph should read really fluid. You should be able to read the paragraph and answer the questions, but not make it obvious, right? So I don't want, it shouldn't be choppy. It shouldn't be obvious that every sentence in your paragraph is going to a question. So you really want to make it flow. You want to embed that answer, like I said. Okay. Any questions about writing a lab report in general? And then I'm about to show you some of the questions that you want answered in your introduction. We're embedding it because it's when you read a lab report that's produced and published through one of the academic journals, they make it so that you get enough information, but it's not like it flows so that it all makes sense, that it all connects, and that the transitions are well done. So that's kind of what I'm saying. They're going to give you questions to address, but it just doesn't sound well. It doesn't sound good. If you were to read a paper and it would have been, if the sentence was like, here, let me go to that slide so I can give you an example. Okay, so in your conclusion, if you were to follow these, if you were to read it, right? And it says, what did you use for controls in your experiment? Let's just say I used temperature and time as a control. Sources of error were I didn't stop it at the same time. I modified or improved this experiment by doing it at the same time. It just doesn't sound very well written. It doesn't sound professional. And I really want to push you guys to, be writing this at a higher level. You guys are capable of doing it and holding you to higher standards of high school, right? Because you guys are ninth graders. So remember, I'm not just looking for the answer. I'm always looking for, I'm also looking for quality and professionalism, because that you guys are at that age where you can and should be giving me that level of quality. So that's kind of why it's not okay to just answer it. I mean, if you were to just answer it, you're looking at least a C or a B, right? But if you really want to get that A, that means that, and remember, the A and the B's and the C's are representative of your learning. So it's not just about points. So if you are able to answer the questions in a way that's embedded and flows nicely, that means that you understand the lab a little bit better. You can articulate yourself well. And that means that you're showing and demonstrating A, understanding and A, work. If you were to just answer the questions, I mean, clearly you know the answers, but that means that there's still some room to learn how to articulate yourself. So you're earning a B or a C. So that's kind of why I'm really big on not just answering it, because there's a lot of tools that you could be learning by writing in a scientific way that doesn't just answer, because we're not just here to just regurgitate information. We're here to articulate and include reasoning for it as well. Okay, so here are the 3 questions that you're going to be answering in your introduction. Remember, you're going to be having this flow in your introduction, but the 3 at the very least, as well as all of the other things that I'm going to go over in the rubric in the next couple slides, you're going to want to answer these 3 questions. The first one is, what is amylase? So remember that your introduction is really setting up the stage, and think about it as if somebody's reading this that they don't know what any of this is. So like, I'm going to be grading it. So pretend like I need to know what amylase is. Now you don't need to go into like this super, super gritty neat detail, but You can tell me that amylase is an enzyme that's found in the saliva and it's part of the digestion process, right? Talk to me about how it plays a huge part in that first beginning aspect of the, of the digestion process. The next question is, how can iodine be used to monitor starch digestion? So they go over that a little bit in the lesson, but yeah, tell me about what iodine is used for. We know we've used it as an indicator for starch, but how is it going to play a role in this lab, right? So making sure that you are answering that question. The third question is, what is the purpose of your experiment, right? Introduction is setting the stage. It's giving us a reason. It's telling us this is important because and why you should continue to read on. So really making sure that you're answering or embedding these three questions into your answer. The questions in your conclusion are going to be, you're going to write an evidence-based statement that summarizes the finding of your experiment. What did you use for controls in your experiment? What are some sources of error? And how could you modify or improve this experiment? So this is really your opportunity that if you goof during the lab and you don't have any other materials to restart or don't have the time, this is a great opportunity for you to point out some of human errors that occurred. What went wrong? Or what do you notice? Oh, I should have done this, darn, right? So this is a great opportunity for you to point out some irregularities so that when it comes time for me to grade the lab report, if there's like something really wonky with your data, that you don't get points taken off because you're acknowledging that the data is wonky, you're acknowledging that something occurred, and that's what's important. It's not so much that if you mess up, it's not that, it's that you acknowledge and you address how you're going to improve it for next time. That's the most important, right? This is a learning process. We don't expect it to be perfect every time. Scientists absolutely do not get things right the first time. Actually, a lot of experiments and a lot of products that we have now today is a result of mistakes that happen in the laboratory. So like, for example, Post-it notes, that was a mistake. They were checking the adhesive for a different purpose and they realized it wasn't strong enough, but it was strong enough to hold up a paper. And that was completely by accident. And now we don't even think of a world without Post-its. I know I don't, because I'm like, with no Post-its, what do I do with all of my reminders and to-do lists, right? So remember, mistakes happen in labs all the time, and it's not always a bad thing. Sometimes it's a great thing. So there's nothing wrong with making mistakes as long as you address them, okay? So that's what you're going to want to include in your conclusion. Questions on that and the introduction or the inclusion. I'm going to go over the grading rubric, but I want to give you guys an opportunity to ask questions first. Okay, last little bit and then we are done and you can get going on your Monday. So for the lab report, the rubric is, the entire lab report is 20 points and it's broken up into 4 sections. 1, your introduction. So 5 points is the most that you can get for each one of these, right? 20 points, 5 points each. And then there's a scale, 5, 4, 3, 2, 1. And what I'm looking for in the introduction is your ability to include a clear description of the principles and concepts related to the experiment. So again, are you explaining to me what you're doing? Why is this important? Why should I continue reading on in your lab report? The next section is your experimental methods. So in order for you to get full credit, you need to include a clear description of how the experiment was conducted. When applicable, describe the controls used in the experiment. Then you're going to want to do data and observations. Again, 5 points, 25% of your grade. And it provides data and observations that are easy to interpret. And if the data tables are created, you're going to include that as part of it and that they are appropriately labeled, excuse me. And the last bit is your discussion and conclusion. So answering questions, making evidence-based claims about the results, and then citing data or observations and discuss possible sources of error. And so that's really what I'm going to be looking for in your lab. Now, the rubric that I'm using is attached to the lesson. So that's going to break it down in way more specific. So if you want to have that lab report open while you're writing your lab, great idea. So you can take a look and see, okay, depending on what you're aiming to earn, you can know what you want to include, right? So that's what I have for you. Do you guys have any questions over the lab in general? I know that was a lot of information. So I just want to make sure that you guys are double checking and making sure that I'm going over anything that you need. Remember, this is on Friday. So there is a couple of days from today until Friday. So I understand if there may, if maybe we forgot something. The good news is I'm recording this session and I'll be posting it so you can re-watch it or skip around to the parts that you need. Are there any questions? All right. Well, we got a couple minutes left. So what do you think? Should I do different? I have some spit in there already. Should I add some orange juice and different juices? It depends on when you get to it, Logan. It technically is assigned for Friday, but if you finish stuff early and you want to start on Thursday, that's fine. All right. If there's nothing else, you are free to go. Let's see what happens if I add iodine to my test tubes where I spit into them. All right. I'm going to add 3 drops to each one. All right, let's see.
I know that I arrived kind of late, but what overall is this lab supposed to be on?
I'm not sure.